Everyone. The subject of today's lecture is energy transformation. That is, how do we utilize the energy that we get from our foods to power all of our energy needs within our body? So we'll go into a lot more specifics as we address individual macronutrients later in the course, but for today we're going to focus on an overview of the process of oxidation of fuels and how it actually works to help us produce the energy we need in our bodies. Okay, so we need energy for just about everything that we do in our bodies. Walk, think, write, build immune cells, active transport systems, pretty much anything that you can think of. And we get this energy from the fuel we consume. That is carbohydrates, lipids, proteins, and ethanol. Now, the primary source of storage is ATP, and the molecule is shown here. So we have an adenine, we have a ribose, and we have a, in this case, triphosphate, TP. So these three phosphates are really the source of where all of our activity is going to occur for energy storage and energy utilization. And these bonds between these phosphate groups are very high energy. So because they're bound here, they really store this energy in their bonds so that when it's broken apart, it's released and we can utilize the energy. So ATP is primarily generated through cellular respiration, that is the oxidation of our fuels to carbon dioxide and water, and it's the major energy supplier in metabolism. Now, this is a really neat molecule because it helps serve as an energy reservoir in our bodies. So like I said, we have these high energy bonds, and it's just waiting there for when we need it. And when we do, it can enzymatically break apart, so enzymatic hydrolysis of the anhydride phosphate bonds, and that releases this energy that we need to do the work that our body requires. So ATP is a primary intermediate for energy releasing and demanding chemical reactions in the cell. Now, when a phosphate group is removed from ATP, we're left with ADP plus an inorganic phosphate. And this phosphate can be added to another substrate or molecule, and then that bond becomes high energy. And that's going to be important as we talk in our talks coming up. So this is about as simple of a schematic as you can get for the ATP-ADP cycle. So we can see on top we have ATP, which contains stored energy. We need the energy for the work that our cells need to do, and then it's broken, so the energy is released, and we're left with ADP, an inorganic phosphate. So we don't have all of this stored energy in this bond here. Then we consume energy from food, and it allows us to rebind this ADP and phosphate so that, again, we have this stored energy for when we need it. Now, how is ATP formed? Well, counterintuitively, it begins with phosphorylation. So that is the enzymatic transfer of an ATP, phosphate from an ATP to another molecule, phosphorylation. Transfer of the phosphate. Now, this might be counterintuitive because you're thinking, I thought we were supposed to be making ATP, but it really takes this initial investment of phosphorylation. Now, this reaction has two steps. First, the lysing of the ATP to the ADP plus the phosphate, then the addition of this phosphate onto a different substrate. So one example of this that's really relevant to our class is the addition of the phosphate onto glucose. Now, enzymatic phosphorylation of glucose by ATP is the first reaction glucose undergoes upon entering the cell. Now, when the ATP is broken, it lends its phosphate to the glucose. So then we have ADP left over, and the high energy phosphate bond is on the glucose, and it becomes glucose 6-phosphate, or G6P, you might have seen it as. And then that contains the high energy bond that can be stored as glycogen, and we can use it whenever we need it. for energy, such as an exercise or when we just haven't consumed energy for a while. Now, the breaking of ATP is highly exothermic, so it releases a lot of energy. And because it releases so much energy, it makes the endothermic reaction of number 2, where we're adding the phosphate onto a different substrate, energetically favorable. So although there is an endothermic reaction, in total, it's exothermic and energetically downhill, energetically favorable. Okay, so then ATP can be formed through 2 main mechanisms. Now, the formation of ATP is highly controlled within the cell to maintain homeostasis of the ATP levels. So the first mechanism is called substrate level phosphorylation. And it's just what we talked about. Transfer of a phosphate group from a phosphate donor to ADP. So it's opposite of the reaction we just talked about. So the phosphate is going onto the ADP to make ATP, and that stores our energy. Now, the main mechanism that is primary for ATP production is oxidative phosphorylation. And just like the title implies, it requires oxygen and produces carbon dioxide, which is called respiration. Now, energy from the mitochondrial electron transport chain, which can also be called the respiratory chain, is used to unite ADP and the phosphate so that we have our energy stored in the form of ATP. So let's pause for a minute and do a little practice while you try to describe the mechanisms involved in ATP formation, both the reactions and the general mechanisms. Okay, so let's talk for just a second about this mechanism of substrate level phosphorylation. Now, there's one especially important reaction that uses this mechanism, and that is conversion to ATP in an anaerobic environment. So we don't have oxygen, but we need ATP. And this can only happen with carbohydrates. We can't do this with proteins or fats. And what occurs is this anaerobic glycolysis, or breaking down of the glucose without oxygen. And this is critical for cells that don't have enough oxygen. Say our arteries are super clogged and we're just not getting enough oxygen, or we're having respiratory problems, or we're just exercising so hard that we don't have enough oxygen to fuel the work that we're doing. So in that case, glucose would be broken to pyruvate, which would normally go to the TCA cycle, an electron transport chain, but instead it goes to lactate. And that's why when we're like totally out of shape and we work out and our bodies aren't very good at utilizing the oxygen we're getting, we get this buildup of lactic acid. However, we can aerobically train ourselves to utilize our oxygen better. Okay, so now on to the major topic of this talk, which is the TCA cycle and oxidative phosphorylation. So all fuels can be used for aerobic conversion to ATP, that is, in the presence of oxygen. So let's take the example of glucose again, because it's just the most common that we think of. So glucose undergoes glycolysis, and then it produces pyruvate, which can enter the inner mitochondrial membrane and be converted to acetyl-CoA, and enter the TCA cycle for ATP production. And this acetyl-CoA substrate is very important to remember because it's the common substrate that we need from all the different types of fuels in order to power the TCA cycle and electron transport chain. So here's just a very rudimentary little graphic that shows glucose is converted to acetyl-CoA, as are amino acids and fatty acids. These go, in the presence of oxygen, these go to the TCA cycle, the electron transport chain, and this creates energy. If we were anaerobic, we would stop here at pyruvate, which would be converted to lactate. So the TCA cycle produces over 2/3 of the energy produced from fuel oxidation. And again, the substrate is acetyl-CoA from the fuel oxidation. So here is our beautiful TCA cycle. And let's just kind of go through it quickly. We'll come back to it a little bit later. But you can see we have a number of steps. And what's happening here is that as one substrate is transformed into another substrate, it's being oxidized and reducing, at different steps, reducing these electron acceptors so that they can go to the electron transport chain and create energy. Now, the small type here is the enzymes that power these conversions. And we notice that where we have these electron acceptors, NADP, NAD, FADH, we also have these dehydrogenase enzymes, and that's really important to remember. Now, you might get confused. Some of these might just say NAD instead of NADP. For the purposes of this class, they're the same energetically, NAD and NADP. They just differ by one phosphate group, but it's not really where the action is. So they do require different cofactors and enzymes, but again, for the purposes of this class, they're energetically similar. Okay, so reminder, what is a dehydrogenase reaction? We talked about it last week. Dehydrogenases transfer hydrogen from a substrate to an electron-accepting coenzyme, such as NAD. So here is two reactions that we see in the TCA cycle. You can go back and see where they are. And we see that first we start with malate. Malate donates its hydrogens to this electron acceptor. So it is, in the process, NAD is reduced because it accepts the electrons and the hydrogens. And malate is oxidized because it loses the electrons and hydrogens. And then NADH can go to the electron transport chain to create energy. And we have the same thing in this step over here. Succinate donates its electrons and hydrogens to FAD. Therefore, it reduces FAD, and it itself is oxidized. And notice these are both dehydrogenase enzymes. So I'm going to call this part 1, and we will resume at the next slide.